she's still going. She's going the whole way. She doesn't need any help. She's over.
Ali Miller with the handoff. And Miller scores for Ireland.
When you first hear the word rugby, the first thing that comes to mind, it's players getting hit, tackling, its aggressiveness, or its similarity to football. Rugby originated in 19th century England, where it was originally a game between soldiers on faraway battlefields. In comparison to football, rugby rules are much more complex than football and require greater skill and endurance. Although there are many fun aspects of playing contact sports, there are some risks that we need to take into consideration. Chronic traumatic encephalopathy, also known as CTE, is a progressive and fatal brain disease associated with repetitive traumatic brain injuries, including concussions and repetitive blows to the head. According to New Switzerland journalist Ben Heather, about 1,200 people suffer head injuries while playing rugby each year. Symptoms of CTE include memory loss, impulsive control problems, aggression, depression, anxiety, suicidal thoughts, and eventually progressive dementia. This disease has so much effect on players that former NFL player Aaron Hernandez, who died in prison at the age of 27, reportedly had a brain so damaged that it was akin to a 60-year-old brain. Currently, there is no cure or treatments for CTE and no conclusive way to diagnose it while a person is alive. Diagnosis requires evidence of degeneration of brain tissue due to deficiency of acetylcholine, or evidence of deposits of tau protein, which are proteins that help stabilize the internal skeleton of nerve cells in the brain. There are certain medications that are currently used to manage behavioral and cognitive problems. Galantamine is a drug that manages memory loss symptoms. This is galantamine's chemical structure. Galantamine is a tetracyclic skeleton with 2 orthoaromatic protons in the ring. Orthoaromatic protons in a ring, illustrated in the right, are when 2 substituents, A and B, are in close proximity, which are shown by the pink dots in galantamine's structure, which is shown in the left. Here we can see a tertiary amine, labeled as blue, a hydroxyl group at the left bottom, and an ether group on the top left. Due to galantamine's rings, galantamine is mainly considered a hydrophobic molecule. Now we will take a closer look into nicotinic acetylcholine receptor's structure, mechanism, its normal role in the brain, and its role in CTE patients when bound to galantamine. In this picture, we can see the different lobes of the brain. Because we are looking into the management of memory loss symptoms, we will be focusing in the temporal lobe and the hippocampus. Nicotinic acetylcholine receptors are also found throughout the brain in parts of the cerebellum and the thalamus. As seen in the past illustration, neurons communicate through a combination of chemical and electrical impulses. In this drawing, we can see acetylcholine, a neurotransmitter responsible for memory function, which is labeled as blue. Acetylcholine is released to the postsynaptic cleft, the bottom section, when an action potential illustrated by the yellow and orange bolts reaches the presynaptic cleft. Normally, to keep a balance of acetylcholine, acetylcholinesterase, the green-looking Pac-Man, breaks down acetylcholine into acetate and choline, as shown by the arrows in the middle of the pre- and postsynaptic cleft, also known as the synaptic cleft. Fun fact is that choline is again recycled to make more acetylcholine when it is bound with an acetyl group. Acetylcholine acts on muscarinic and nicotinic receptors. Muscarinic receptors are also found throughout the brain, but play a role in cardiac inhibition by decreasing heart rate and reduction in heart contraction force. Another fun fact is that just like nicotinic acetylcholine receptors name portrays, nicotine can also bind to the receptor. This makes nicotine and acetylcholine agonists. When galantamine is bound to acetylcholinesterase, it acts as a reversible and non-competitive inhibitor by binding to the allosteric site of the receptor, which is seen in the presynaptic cleft. The binding leads to a conformational change that increases the receptor's response to acetylcholine. This context is also explained in the middle drawing, where the birthday hat galantamine is on top of the acetylcholinesterase, the green Pac-Man, inhibiting the breakdown of acetylcholine, which results again in the increase of acetylcholine. Some side effects of galantamine include nausea, vomiting, diarrhea, decreased heart rate, and weight loss. In this 360 illustration, we can see how nicotinic acetylcholine receptor is a quaternary structure that consists of more than one subunit. The receptor has 5 subunits, which are labeled in different colors. These 5 subunits consist of 2 alphas, 1 beta, 1 gamma, and 1 delta, and assemble in both heteromeric and homomeric pentamers. It can be a homomeric pentamer because it contains 2 similar alpha subunits. These receptors are structurally similar. As seen in the top left, each include a long extracellular N-terminal domain, 4 transmembrane segments, M1, M2, M3, and M4, a cytoplasmic loop of variable length, and an amino acid sequence between the 3rd and 4th transmembrane segments, and a short extracellular C-terminus as shown in the bottom right of this picture. In the top of this picture, we can see how the N-terminal domain contains amino acids that are delimited by 2 cysteine residues that form a disulfide bridge. This is the cis loop that defines all subunits in this receptor family. This cis loop is also believed to be important in the transduction of agonist binding into channel opening. Nicotinic acetylcholine receptor is a ligand-gated ion channel protein composed of 3 domains: extracellular, transmembrane, and intracellular domain. For channel to open, acetylcholine must be bound to the binding site. The opening provides potassium efflux and sodium influx. In the part B of this picture, we can see the competitive binding pocket where the receptor contains 5 aromatic residues and is bonded to cysteines. In part C, we can see other amino acid residues such as tyrosine and tryptophan. Something to notice about this picture and upcoming pictures is that each subunit has its own secondary structure. In the right, we can see how beta helices gather in the extracellular hydrophilic side, labeled as ECD, and are mainly antiparallel, and alpha helices gather in the transmembrane and intracellular hydrophobic side. This video closely shows the secondary structure in the receptor. The ball and sticks shown are bound galantamine molecules, which I will get into more deeply. The blobs shown are galantamine. There are 5 galantamine molecules bound to one receptor, and each galantamine is found between 2 protein subunits. This is a closer illustration. Now we will talk about some of the dominant non-covalent interactions that occur between the nicotinic acetylcholine receptor and galantamine. Most of the interactions happen between galantamine and tyrosine and tryptophan residues, which are labeled in purple on the left. Galantamine interacts with tryptophan through pi stacking between benzene rings, and through cation pi interactions between the benzene ring and the nitrogen cation in galantamine. While tyrosine binds with galantamine through pi-aq bonds and carbon-hydrogen bonds. In the carbon-hydrogen bond, tyrosine's hydroxyl group is the acceptor, and the carbon of galantamine acts as a donor. On your right is a summary of some of the main interactions that were just mentioned. Here we can see a 360 view of the binding site of galantamine in green, interacting with mostly tyrosine 188, 195, 93, and 55, and tryptophan 147. Where do we go from here? How do you prevent CTE? Do you just stop playing contact sports? Especially a sport so ingrained in our society. Maybe you don't stop at all. Currently, there has been petitions to the NFL to ban the use of helmets, and there are now players who play football without helmets to avoid more head injuries. Because people are already playing football without helmets, is it safe to play rugby as well? Maybe we can just play these sports in a bubble, like in the sport called bubble soccer. Sounds fun, right? Or maybe we can use these automatic bubble head activators called Hubden, which were invented by a Swedish safety technology company for urban cyclists. There is still so much more that we need to understand about nicotinic acetylcholine receptors and their role in memory. For example, why there are 2 natural agonists, acetylcholine and choline, that bind to alpha subunit, and how to identify the conditions under which agonists would play a major role. And is this a way by which selective activation or inactivation of a particular nicotinic acetylcholine receptor subtype occurs? There are still many questions that remain unanswered. But again, how do you think we can prevent CTE?